1.2 million people in Germany suffer from Alzheimer's disease, and the number continues to rise. On the left, a healthy brain. On the right, that of a person with Alzheimer's disease. Alzheimer's disease is thought to occur when clumps of protein called plaques form in the brain. The plaques, coloured green, prevent information being passed on to the nerve cells coloured red. But why? Franz Ulrich Hartl and his team at the Max Planck Institute of Biochemistry in Martinsried near Munich want to find out. They are investigating what makes proteins form these clumps. Professor Hartl has made a far-reaching discovery: the important role of the chaperones. These are molecules which help proteins to find the correct form. He is a pioneer in this field.
Proteins stick together because they are not correctly folded. They expose sticky side chains, which then stick together, like these two lumps of dough. They cannot release themselves again independently. But that is exactly what we want to achieve, in order to prevent neurodegenerative diseases.
This is how plaques are formed in the cell. Along the ribosomes, a chain of amino acids is formed. These are the primary structures of every protein. The protein is long, thin and sticky like a roll of dough. However, proteins are only useful for cells when their chain of amino acids folds into a particular shape, like this pretzel. Or Sopf here, for example. However, many proteins cannot fold themselves without help, just as the dough cannot find its correct shape without the baker. Within the cell, the chaperones take on this task. To do so, they need energy in the form of ATP synthase. Without chaperones, the proteins, or the pieces of dough to stick to our image, will not find the correct form and will therefore not be biologically active. An incorrectly folded protein is usually sticky and glued to itself, or glues itself to other proteins. This makes it useless for the cell, junk, so to speak. And the dangerous plaques are formed. Now the chaperones adopt their second role and function as controllers. They recognize the junk and collect up the useless proteins. Then they have them chopped up in a special shredder, the proteasome, so that parts of them can be recycled later.
I want to know how we can increase the number of chaperones in the cells so that they can prevent the formation of dangerous plaques, which collect like sticky dough around the nerve cells and restrict their function. For that, we need to understand what and who is involved in the process, so that we can use the cell's own weapons to overcome the disease.
In this case, the cell's own weapon is its emergency reaction to stress. Cell cultures are exposed to heat, and the proteins in the cell are denatured. This means that their biologically active structure is lost and has to be restored. The response to stress is that increasing numbers of chaperones are formed, a fact which scientists can prove by means of gel electrophoresis. Indeed, chaperones, also known as heat shock proteins, or HSP for short, were discovered in the first place through this effect. Now they can be seen as blue strips within the gel. Professor Hartl and his team want to know whether this stress response can also be produced in a living organism, the roundworm Caenorhabditis elegans, and whether it is possible to specifically increase the production of this important folding aid.
I want to know whether there are chemical substances which trigger the same reaction as the heat stress, namely the increased production of chaperones.
In their search for a chemical substance, the scientists came across geldenomycin. It bonds specifically to the heat shock protein HSP90 and changes its behavior within the cell. HSP90 proteins play an important role in the survival of the cell. But exactly how does geldenomycin bond with HSP90? And can it really increase the production of chaperones? The scientists use a mass spectrometer to measure whether the quantity of chaperones has been increased by the geldenomycin. It can identify elements within the cell and determine their mass, including the number and type of chaperones. The result? If geldenomycin is added to the chaperones, they are poisoned, so to speak, because geldenomycin blocks the place where ATP would normally bond. The HSP90 chaperones flake out. That stresses the cells and they start up their emergency program. Their immediate response to the stress is to produce a variety of different chaperones, including HSP40, 60, 70, 90 and 100. The protein folding machinery is boosted enormously. Together they take over the tasks of the blocked chaperone. Indeed, because several bakers can produce more than a single one, more proteins are also produced.
With this experiment, we were able to prove that chemical substances like geldenomycin really do boost the chaperones, in the same way as the heat shock response.
That sounds like the longed-for solution to a huge problem. But can geldenomycin also help with neurodegenerative conditions like Alzheimer's disease? To test that, the scientists changed some of the genes in the roundworm. They allow certain proteins which are responsible for movement to strongly stick together. And so that they can observe exactly what happens next, the scientists mark the plaques with a green fluorescent protein known as GFP.
Imagine that this lump of dough is a protein plaque. And now we have marked it within the cell with fluorescent molecules, as if this light bulb were stuck inside the lump of dough.
The genetically altered worms can hardly move because of all the plaques in their muscle cells. We can identify the agglutinations or clumps by the illuminated dots. And now the experiment proper can begin. The scientists treat some of the worms with geldenomycin in order to trigger off a stress response and increase the production of chaperones. Indeed, the roundworm, which was previously sick, can now move again and appears to be healthy. It is constantly on the move in search of food, in contrast to the untreated worm, which is still lying there completely stiff and motionless. We can also see this through the marking of the plaques with GFP. The plaques have mostly been destroyed on the left, but not on the right. The experiment could be a breakthrough in the treatment of neurodegenerative diseases, because geldenomycin ensures that more chaperones are produced and the plaques destroyed. But the apparent stress medicine has a downside.
Unfortunately, the problem with geldenomycin is that it is too toxic to be used in humans. It is not suitable for use as a medicine, at least for long periods. So we shall have to carry on searching for suitable chemical substances.
Not all the substances that speed up the chaperone system have been identified yet. Professor Hartl is sure that we can find suitable substances to activate human chaperones, so that one day these may be able to cure patients with Alzheimer's, Parkinson's and Huntington's disease.